Hello everyone, my name is Rabluti and this is the beginning of chapter 2, which is all about the fundamental units of life, aka the cell. And cells are as fundamental to the living systems of biology as the atom is to chemistry. And as you know, all organisms are made up of cells, and the cells are the simplest collection of matter that can be considered as a living entity. And indeed, many forms of life exist as single-celled organisms like this paramecium, but organisms that are more complex are multicellular, like you and I. And so all cells are related by their descent from earlier cells. And as you might know, during the long evolutionary history of life on Earth, cells have been modified in many ways. And this modification is happening every day as well. And although cells can differ substantially from one another, they share some common features. And so these are the main subjects for this video, and so let's go learn more about cells. So before we start learning about cells, we first have to know how biologists even study cells. So the two main ways that cells are examined are by using biochemistry and by the help of microscopes. Okay? So before we start learning about cells, we first have to know how biologists even study cells. So the two main ways that cells are examined are by using biochemistry and by the help of microscopes. Okay, so let's talk more about microscopes. So the development of instruments that extend the human's senses allowed the discovery and early study of cells. And so microscopes were discovered in 1590 and further refined during the 1600s. And the cell wall was first seen in 1665 as Mr. Robert Hooke was looking through his microscope and examining the bark of an oak tree. But it wasn't until Antonio van Leeuwenhoek that had crafted lenses that could help scientists see living cells and not just the cell wall. And so the microscopes that we see in labs are mostly light microscopes, and they are called light microscopes because visible light passes through the specimen and then it reaches our eyes. And so by bending the light that passes through the microscope, the specimen is magnified. And the three most important pa parameters of the light microscopes are: number one, its magnification; number two, its resolution; and last but not least, number three, its contrast. Okay, so magnification is the ratio of an object's image size to its real size. And so the magnification of light microscopes, a light microscope is about 1,000 times the actual size of the specimen. And have in mind that as magnification increases, the clarity of the image decreases drastically. And so the resolution of a microscope refers to the clarity of the image, or it is the minimum distance between 2 points that can be separated and still be distinguishable as separate points. And the light microscope can distinguish 2 points uh, that have a distance of 200 nanometers regardless of the magnification. And so the third parameter is contrast. Okay, and I'm sure those of you who do photo editing and such have seen this one. So, as you might know, this is the difference between the light and difference in brightness between the light and dark areas of an image, and the of methods for enhancing the contrast include staining or labeling cell components to stand out visually. And until recently, the resolution barrier prevented cell biologists from using standard light microscopes when studying organelles. And to see these structures in any detail required the development of a new instrument. So in the 1950s, the all-new and powerful electron microscope was introduced to biology. The main difference between the light microscope and the electron microscope was that rather than focusing light or photons, the electron microscope focused a beam of electrons through the specimen or onto its surface. And it's very important for you to know that the resolution of the microscope is inversely related to the wavelength of the light or electrons that the microscope uses to give us a picture, and electron beams have much less wavelength than visible light. So thus electron microscopes have very better resolution in their pictures. And modern electron microscopes can theoretically achieve a resolution of up to 0.002 nanometers, though in practice they usually cannot resolve structures smaller than about 2 nanometers across. But still, this is a hundredfold improvement over the standard light microscope. And so another kind of microscope is the scanning electron microscope, or uh, also known as electron microscopes. And micrographs that are taken by these microscopes, show a 3D image of the surface of the specimen. And so here's a picture of a cell from a trachea which is covered with cilia. And as you might know, the pictures taken from electron microscopes are black and white, but as you can see in these pictures, they have more colors. And that is by the help of computers and AI. And there's another kind of an electron microscope, and it's called a transmission electron microscope, or aka TM electron microscopes, which the pictures taken from these microscopes are a thin section of a specimen. And here's the same picture that I showed you, but from a transmission electron microscopy. And I know the other picture was way cooler, but using this microscope we can see the inside of cells, not just the surface. And have in mind that the stain that we have to use in TM microscopes must be a heavy metal, and for scanning electron microscopes we have to use a thin layer of gold on the specimen. And electron microscopes have revealed many subcellular structures that were impossible to resolve with the light microscopes. But the light microscopes offer advantages of their own. For example, the ability to study living cells. But in the methods used to prepare the specimen in electron microscopes, in all electron microscopes, kill the specimen as well. So yeah, sacrifices must be made. Okay, and so microscopes are the most important tools in cytology, or the study of cells, but to understand the function of each structure, however, requires the integration of cytology and biochemistry, aka the study of chemical processes of cells. And so that was all for this video, and as always, until the next video, happy learnings to you all.